Imagine a world where critical health markers, environmental pollutants, or agricultural needs are continuously monitored without human intervention. This is the promise of autonomous biochemical sensing, a revolutionary field at the intersection of biology, chemistry, engineering, and data science. It's a frontier ripe for exploration by scientists, engineers, researchers, and students alike. Autonomous biochemical sensing refers to sophisticated systems designed to detect and analyze biochemical substances. These include vital molecules like ions, proteins, DNA, metabolites, or harmful toxins. The defining characteristic is their ability to operate independently, generating high-resolution data without direct human oversight. These systems are transforming how we gather information in diverse fields. From personalized healthcare to global environmental monitoring, their impact is profound. They offer unprecedented access to real-time granular data, enabling proactive decision-making and scientific discovery. The drive towards autonomy in biochemical sensing stems from critical limitations of traditional methods. Manual sampling and laboratory analysis are often time-consuming, labor-intensive, and prone to human error. They also provide only snapshots in time, missing dynamic changes. Autonomous systems overcome these hurdles by offering continuous, remote monitoring capabilities. This is crucial for applications requiring immediate responses, such as detecting rapidly spreading pathogens or sudden environmental contamination. For researchers, it means access to vast, uninterrupted datasets for deeper insights. At its heart, an autonomous biochemical sensor integrates several key components. A recognition element selectively binds to the target analyte, initiating a measurable change. This change is then converted into an electrical signal by a transducer. Sophisticated signal processing and data analysis algorithms interpret these signals. Finally, communication modules transmit the data, often wirelessly, to a central system for storage and further analysis. This intricate interplay enables the sensor's independent operation. Despite their immense potential, the development of robust autonomous biochemical sensors faces significant scientific and engineering hurdles. Addressing these challenges is paramount for widespread adoption and reliability. Each presents a unique research opportunity for innovation. One of the most pervasive challenges is biofouling, where non-target biological molecules adhere to the sensor surface. This unwanted accumulation can block recognition sites, leading to reduced sensitivity and accuracy over time. It's a major impediment to long-term deployment. Research efforts focus on developing anti-fouling coatings and surface modifications. These include zwitterionic polymers, self-assembled monolayers, and nanostructured materials designed to repel non-specific adsorption. Understanding surface chemistry at the nanoscale is critical here. A sensor's selectivity refers to its ability to specifically detect the target analyte amidst a complex mixture of similar molecules. In biological samples or environmental matrices, numerous interfering substances can lead to false positives or inaccurate readings. Achieving high selectivity is a fundamental requirement. Strategies to enhance selectivity involve engineering highly specific recognition elements, such as aptamers, molecularly imprinted polymers, MIPs, or genetically modified receptors. Advanced signal processing techniques can also help differentiate target signals from background noise. This area demands deep understanding of molecular interactions. Sensitivity defines the lowest concentration of an analyte that a sensor can reliably detect. Many critical biomarkers or environmental contaminants exist at extremely low concentrations. Therefore, high sensitivity is often crucial for early detection and effective monitoring. Improving sensitivity often involves signal amplification strategies. These include using nanomaterials to increase surface area for binding, employing enzymatic reactions to generate amplified signals, or integrating optical and electrochemical amplification techniques. Miniaturization and noise reduction are also key factors. Current autonomous sensors are often designed for a narrow range of specific analytes. This limits their utility in scenarios requiring broad-spectrum analysis or the detection of unknown compounds. Expanding this range is vital for comprehensive monitoring. Developing multi-analyte sensors or sensor arrays is a promising approach. These systems integrate multiple recognition elements, each tuned to a different substance. Machine learning algorithms can then interpret the complex signals from these arrays to identify various compounds simultaneously. Reliable sensor operation requires periodic calibration to ensure accuracy and compensate for drift. Performing this autonomously without human intervention is a significant engineering challenge. Manual calibration is impractical for remote or long-term deployments. Research is exploring on-chip calibration mechanisms, such as integrated microfluidic systems that deliver known standards. Machine learning models can also be trained to predict and correct sensor drift based on environmental factors or historical data. This requires robust algorithms and precise fluidic control. The operational lifespan of autonomous sensors is often limited by factors like recognition element degradation, battery life, or sensor fouling. For applications requiring months or years of continuous monitoring, extending this lifetime is a critical design goal. This is particularly challenging in harsh environments. Innovations in materials, science, power management, and self-sustaining energy harvesting are crucial. Developing robust, stable recognition elements that resist degradation is also a key research area. The goal is to create fit-and-forget devices that operate reliably for extended periods. To overcome these formidable challenges, current research strategies involve a paradigm shift, rethinking traditional approaches and embracing interdisciplinary solutions. This includes drawing inspiration from nature and leveraging cutting-edge materials, science, and fabrication technologies. Moving beyond conventional sensor designs involves integrating advanced microfluidics, novel transducer materials, and sophisticated data analytics. This holistic approach aims to create more robust, versatile, and intelligent sensing platforms. It requires a deep understanding of the entire sensing pipeline. For instance, integrating sample preparation steps directly onto the sensor chip can reduce external interference and improve overall system performance. This lab-on-a-chip concept is central to next-generation autonomous systems. It streamlines the analytical process significantly. Nature provides an unparalleled blueprint for robust and adaptive systems. Biomimetics involves emulating biological processes and structures to design more effective sensors. This includes mimicking the highly selective recognition mechanisms found in biological systems. Examples include developing artificial noses that mimic olfactory receptors for broad-spectrum chemical detection. Another area is creating self-cleaning surfaces inspired by lotus leaves to combat fouling. This interdisciplinary field combines biology, chemistry, and engineering. The development of novel materials is fundamental to advancing autonomous sensing. Advanced polymers, nanomaterials, and smart hydrogels offer unprecedented control over sensor properties. These materials can enhance sensitivity, selectivity, and stability. For example, graphene and carbon nanotubes provide high surface area and excellent electrical conductivity for enhanced signal transduction. Stimuli-responsive polymers can be engineered to change properties in response to specific analytes, enabling new sensing modalities. Additive manufacturing, or 3D printing, is revolutionizing sensor design by enabling the creation of complex, multifunctional platforms. These technologies allow for precise control over sensor architecture, integrating multiple components and functionalities into a single device. 3D fabrication facilitates the creation of intricate microfluidic channels, varying surface polarities, and tailored hydrophobicities. This allows for optimized sample delivery, enhanced analyte capture, and improved signal generation. It opens new avenues for miniaturization and integration. A key development is the integration of biomimetic functions like self-diagnosis and self-repair capabilities. Inspired by biological systems' ability to maintain homeostasis, these features aim to extend the sensor's lifespan and reliability in challenging environments. Self-diagnosis involves onboard monitoring of sensor performance and identifying potential failures. Self-repair mechanisms, such as the release of encapsulated healing agents or reversible binding sites, can restore functionality after damage or degradation. This is a frontier of materials, science, and robotics. Artificial intelligence and machine learning algorithms are indispensable for processing the vast amounts of data generated by autonomous sensors. They can identify complex patterns, compensate for sensor drift, and improve the accuracy of analyte identification. These algorithms can learn from historical data to predict sensor behavior, optimize calibration routines, and even detect anomalies indicative of sensor malfunction. This intelligent data processing transforms raw signals into actionable insights, enhancing the autonomy of the system. To overcome the limitation of detecting a single substance, researchers are developing multimodal sensors that combine different sensing principles, e.g., electrochemical, optical, mass-based. Sensor arrays comprising multiple individual sensors provide a richer dataset. By integrating diverse sensing modalities, these systems can provide more comprehensive and robust information about the sample. The redundancy and complementary nature of the data improve reliability and reduce false positives, especially in complex matrices. The transformative potential of autonomous biochemical sensing spans numerous critical sectors, promising to revolutionize how we monitor, diagnose, and manage various aspects of our world. In healthcare, autonomous sensors enable continuous, non-invasive monitoring of vital biomarkers, facilitating early disease detection and personalized treatment. Wearable sensors can track glucose levels, cardiac markers, or stress hormones in real time. This empowers patients and clinicians. These systems are crucial for remote patient monitoring, especially for chronic conditions, reducing hospital visits and improving quality of life. They also hold promise for rapid point-of-care diagnostics in resource-limited settings, democratizing access to critical health information. Autonomous sensors are vital tools for safeguarding our environment. They can continuously monitor air and water quality, detecting pollutants like heavy metals, pesticides, or industrial chemicals. This provides immediate alerts for contamination events. Deployment in remote locations, such as rivers, oceans, or industrial sites, allows for comprehensive environmental surveillance. This data is essential for regulatory compliance, ecological research, and informing public health interventions. In agriculture, autonomous sensors can optimize resource use and improve crop yields. They monitor soil nutrient levels, moisture content, and plant health indicators. This enables precision agriculture, reducing waste and increasing efficiency. These sensors can detect early signs of plant disease or pest infestation, allowing for timely intervention and minimizing crop loss. This contributes to sustainable farming practices and global food security, a critical challenge for our growing population. Ensuring food safety and quality is paramount. Autonomous biochemical sensors can detect pathogens, spoilage indicators, and adulterants in food products throughout the supply chain. This prevents foodborne illnesses and maintains product integrity. From farm to fork, these sensors provide continuous monitoring, ensuring compliance with safety standards and reducing waste due to spoilage. They offer a rapid, non-destructive alternative to traditional laboratory testing, enhancing consumer confidence. Hey, if you're enjoying this video and want to see more like it, don't forget to hit subscribe and turn on the notification bell so you never miss an update.